thanks, David and everybody else for the invitation to this spectacular place, and also LMB for the great hosting. And yeah, I'm talking about, let's say, with the topic of how significant this is to the genome, we going to the regime of maybe 10 to the minus 7% or so. Basically, DNA breaks, which we, thank God, have very rarely. And these breaks are induced by ionizing radiation, this everybody knows, but we also have certain genotoxic chemicals, topoisomerases, endogenous processes, for instance, doing replication or transcription, as we just heard. Replication itself is a very prominent source. We get a lot of so-called single-ended breaks, but also we have programmed DNA breaks in the immune system for the antibody diversity and in meiosis. So for basically the germline rearrangements of the genome. Now, one of the core factors that these breaks are detected is the so-called MRN complex that together with the Q protein, which is a ring-shaped protein, as you see here. This is a Q end binding factor, and this MRN complex is a more complicated machine. It's basically an ATPase and a nuclease that then recognizes these breaks, and then basically the cell, especially since breaks are so problematic for the genome stability, kind of enter in sort of a panic reaction almost, and they kind of start to tether these breaks, and they recruit a large kinase called ATM. And this kinase is a very elaborate auto-inhibited machine, and this becomes then activated at these breaks by a mechanism we still don't really understand structurally. And what happens is that this kinase phosphorylates a lot of targets, many of which are chromosome-binding proteins, repair factors, and so forth. This leads to kind of rearrangement of chromatin, a lot of, let's say, post-translation modifications going on, basically omit a lot of steps here. Eventually, this kind of results in a complex called shelterin that is assembled in these chromatin regions and basically shuts down transcription in these areas and basically protects the genome for erosion through nucleases and aberrant transcription and these kind of processes. So so basically these are shielded, and then another repair process called anonymous end joining, where basically ligate these ends together. This is rather promoted in this kind of environment. Now, when we enter G2 phase and S phase in human cells, so we have, let's say, a sister chromatid around them, and this enables the cell to more accurately repair this by so-called homologous recombination. There's kind of a logistic switch in the cell through basically cell cycle dependent kinases. They phosphorylate then other factors like BRCA1, BART1 complex, a very well-known protein, and a very purely characterized protein called CtIP, which is a very elongated protein. And they are phosphorylated and basically primed through the cell cycle, but then if you have the DNA damage response, additional phosphorylations take place, and then they assemble in a different structure around these break sites, and this generates then a more chromatin that is believed to be amendable for homologous recombination. So this is the so-called repair foci. We know when we irradiate cells, these are very large, gigantic structures of assemblies form here. And current models indicate that such a foci, you have these kind of domains with the BRCA1 more in the center and this kind of shielding parts more in the periphery in these loop regions. Now then chromatin remodeling, enters a variety of different remodelers are thought to play important roles here. This is one from our laboratory, one of these remodeling complexes. Eventually you clear the DNA, and then this MRN complex is still in an inactive state. So as I mentioned, it's like an ATPase and a nuclease, and what it does, it needs to cleave the DNA to basically generate a free DNA end that then can be used in homologous recombination. And this is triggered by the CtIP protein, which is kind of an activator. And it's also a scaffolding protein, and then you have this nuclease activation of the mRNA complex. And so if you zoom in here, so the mRNA complex, this is the Q, here would be the broken end. It will basically cut the 5' strand as seen from the end here. And then we may have some back resection, then generate a little bit single-stranded DNA, and this is then an assembly platform, which I don't show here, for, let's say, further complexes that then resect the DNA, generate long single-stranded DNA, and then trigger this repair event. Now, if you look at these mRNA complexes, these are very kind of interesting proteins. They have like a globular part, an ATPase, and a nuclease, and then they're very long, depending on the organism, the species here, or let's say phage in this case. These coiled-coil regions, which we believe are like in this kind of open states where the two coiled-coil arms are separated. They are joined here by a kind of a zinc hook region, so this is a little metal binding site here. And they can also have this kind of more assembled, folded, rod-like states here. Now, what are these coils doing? And one of the things we kind of believe is they are basically gigantic topology filters for DNA. So they basically, the idea that the mRNA complex is a factor that can recognize a very broad range of ends. So meaning like there can be covalently bound proteins sitting there, all sorts of topological structures or so. So it needs to load onto a linear, basically DNA end. And this we've shown in the bacterial system with an experiment where we basically tested whether a blocked DNA end, where normal repair factors cannot simply bind to, with the streptavidin biotin linkage here, can be detected by mRNA in a linear or let's say a recircularized version. So this way basically would probe the topology of this DNA here, and you basically have the same end structures in these substrates. So here's just bound in a double ligation to streptavidin, and here would be free. And this is, so if you assemble this biochemically, this is the species you get on a gel. We have then characterized that further. What we get like, single streptavidin bound, double streptavidin bound, recircularized, and then higher order species. Now, if you add this protein and ATP, you look for survivors. And this is basically the survivor. And basically, if you go to 3 here, this is then basically this part that is untouched. And whatever, if you have a linear DNA, this is basically cleaved away. So early cryo-EM structures we did a couple of years ago. We found basically that this bacterial complex assembles like into this globular head. Here you have the nuclease part, you have the ATPase part, and here you see like an end structure then bound into this complex. It basically forms like a channel that DNA can actually exit here again. And you see this kind of folded large coiled coils. And the interesting part was that there was a single basically opening in these coils where basically a single duplex can go through. Now, why is this significant? So before I come to this, I would like to basically show you some unpublished data where we like kind of filled in some a little bit intermediate steps. So this is the complex when we visualize it in cryo-EM in the presence of ATP gamma S. You see basically this folded head module. You see like these open coil domains. In the cryo we don't see them really. They're kind of averaged out here, but you see that these angles, they open up. The nuclease sits here in an auto-inhibited state. So the nucleus active sites are basically buried here. So this would not be active, this enzyme cleaving any DNA, which is good, obviously. You shouldn't degrade like genomic DNA in this case in E. coli. So then when we add basically again with non-hydrolysyl ATP analog a DNA with, let's say, some end blocks, you can basically observe the same structure here, but now DNA binds into these coiled-coil, in between these coiled-coil regions. The end could be on either side of here. And then we find something very strange in a way, or kind of a miracle occurs when we add ATP, and then we so this is our, let's say, low technology time resource study here. So we add ATP, we incubate it for a while, and then we add beryllium fluoride, which is like a phosphate mimic. And then the idea is that sometime you basically put in, make an ADP beryllium fluoride analog. Then actually what happens is that this M11 part here vanishes. So we get the same structure, more or less the same structure in the Rad50 part, the ATPase, and the nuclease vanishes. So why does it vanish? It's not diffusing away because we still see this attachment to the coils here. I think it's just that these nuclease domains, they probably open up here, so the dimer dissociates, and then these individual parts, they just wobble around, and we average just out in cryo-EM. But what we see here is that these coiled-coils are now clamping, so here they're open, and now they fold into these rod-like states. So in principle, you can visualize this as, let's say, a lot of the folding energy here is, so the coils like to be in the rod state, but you need ATP to kind of open them, and then DNA in some hydrolysis event kind of folds them back together. If you then have only ATP, so no beryllium fluoride, what happens then is that then this part can basically, in a full hydrolysis cycle, reassemble on the other side. It kinks a little bit DNA, and then this nuclease becomes active and then incisors the DNA. So this is basically kind of a opening and refolding or reassembly step on the nuclease, and here would be the structure. So now why is this, I think why is this interesting in terms of topology? Because, I don't know if you see the pointer here, hardly see it anymore, but you have this open state, and if you have a single DNA end, you can basically thread through this DNA, and then you can basically fold this back up, and then you have the single opening where the DNA goes through. Now, if you have, let's say, an internal part of DNA, you can still thread this DNA, bind to the nuclease, to the ATPase, activate ATP hydrolysis, but then this zippering up cannot happen because you have a second DNA strand basically threading through this ring, and this probably would sterically prevent forming of this active state here. This is topologically, I think, quite similar to SMC complexes, which, you know, some of the pioneers sit here in the audience. Cees Dekker and I know Jan Löwe is here in terms of structural biology. So basically, SMC proteins, they have a kind of a related structure, having these coils, and this ATPase is very similar. They have other subunits here. And what they do is they do loop extrusion. So basically, they also engulf kind of this DNA, and then this DNA kind of migrates also through here in a way, and you extrude basically this loop. So topologically, it's kind of a little bit related, but in a way also opposite. So now what is interesting when we move now from the prokaryotes to the eukaryotes is that this prokaryotic complex I just told you about is kind of autoactive. So if you give it DNA ends, it will happily degrade it or cleave it. The eukaryotic complex is very different because if you give it a DNA end, you hardly find any activity. So people have really struggles getting good biochemical activity out of this. And the reason is you need probably a much more elaborate activation event through this kind of CtIP protein. The situation is even more complex because a variety of studies have now shown that this mRNA is actually actively inhibited at our natural telomere ends, natural DNA breaks or DNA ends at telomeres. And there's a factor called TRF2 that basically has a motif that binds to this mRNA complex and seems to kind of inhibit it. And the question, of course, was in what stage this is inhibited. People proposed, or it activates the ATPase, and then this complex bleeds off, but we think that model is not entirely correct. Actually, it's maybe a little bit different. So in order to get to the eukaryotic complex, of course, we played a lot around for years with sample optimization, and finally could also manage a good sample by using certain length of DNA and also having kind of a loading reaction where we first incubate mRNA with ATP. So this hydrolysis step is very important. And then we can stabilize the complex better than with this beryllium fluoride addition, and then image this by cryo-EM. Here's some 2D classes, and then we get a couple of structures. So we could get the complex of MR alone. So this is the nuclease, the ATPase, without NBS1. NBS1 is like a adapter protein that is very important for activating ATM protein, this ATM kinase, and for recruiting some substrates for ATM kinase. This is the MRN complex, the full MRN complex, and we have reasonably good resolution. And we also have a couple of complexes with TRF2 and some TRF2 fragments, both bound to MR, so Mre11-Rad50 alone, and to this full complex. So on the first side, they look all the same, right? And actually, there's not much going on in terms of really the core complex. So here's a more color-coded picture now with the ATPase, here the nuclease. Again, this nuclease is in its out-inhibited state. So normally, if it would be active, it would sit on this side. And we see basically DNA very nicely bound in this clamped state here. Here would be the NBS1 subunit. Same, this is just from the bottom here. And you see that this NBS1 subunit basically wraps around the entire nuclease as if it would stabilize this kind of out-inhibited nuclease form. That's just it. So we can go to more details here. I think that's not so important here. What I think is important is that this kind of very tight zippering up of the coiled-coil region here. So here again, you see this kind of gate, which a single DNA strand can pass. And this part here is very tightly bound, even with some hydrophobic interactions here, forming this very tight out-inhibited clamp. I think this explains very well the structure, that why we hardly see any biochemical activity in terms of nuclease activity. We see DNA-stimulated ATPase activity, but nuclease activity is very, very broad, and we could validate this a little bit with cross-linking mass spectrometry, which also shows a bit that these coils are on other sides. This movie doesn't work very well here. So you might basically say, oh, this looks much like the bacterial complex, but there's actually a very fundamental difference to the bacterial complex. With the bacterial complex, we never actually, in fact, see this state here. In this state, what I mean is we never saw a state, since many years now of studying this, where we have the ATPase in this kind of rod-shaped, zippered up conformation, and the nuclease out-inhibited. So either in the bacterial system, either the coils here are open with ATP, or they are closed, but when they are closed, this Mre11 vanishes. So either we don't see it at all, or it reassembles on the other side. So the human complex kind of decouples this activation, and generates a new state, which we call basically the sensing state, because we think this is very important for the eukaryotic biology of mRNA. So when we look more into this, we also find a very interesting interaction of the NBS1 here, which is this kind of protein that wraps around, and this binds to the Rad50 side. Here there's a beta sheet. This beta sheet is, for the people in the field, is quite famous because there's a lot of mutations have been mapped there in yeast, so-called separation of function mutations that have been shown to be very important for the nuclease activity. And what we see is here that this NBS1 protein binds with kind of a phenylalanine finger into this site and seems to even lock this state down. Now, why is this interesting? Because this motif of this part of the NBS1, which is the very C-terminus, basically, this is quite highly conserved here. So there's some couple of positively charged residues which can bind to the DNA here in the vicinity. But this is a very conserved part. And this motif has been observed before. And this motif has been observed before by the Pavlitic lab bound to the ATM kinase. And when we compare these two structures, here in this intrinsic, let's say, interaction, and here in this extrinsic interaction, we see that the same phenylalanine actually binds to either Rad50 or it seems to bind to ATM, which basically shows us that there needs to be some sort of change going on. So this motif has to dissociate from Rad50 in order to bind to ATM. And despite many trials, so AlphaFold is very useful, but I could not yet get a convincing complex using AlphaFold which kind of fits to, let's say, at least the functional homology we have yet. So I think there is something really interesting going on here, but this we haven't really found out. Now, anyway, so the sensing state, this is just a summary. So here we have this motif sitting here, and then it has to somehow go away. But this complex cannot be, I don't know, I didn't find anything useful with AlphaFold. One can find complexes with AlphaFold, but there's kind of something dramatic going on, which we don't know whether that's true or not yet. So the last part I would like, so the next part I would like to briefly touch upon is the TRF2, which is that this is an inhibitory motif, and this is the TRF2 structure, and we have the entire TRF2 protein, which forms like a dimer, but we only see a tiny piece here, which has been mapped before as the important binding site, and this is actually mapped by AlphaFold spectacularly well. You don't need to change basically a single amino acid to dock this. Here, this is again the cryo map. Here is this TF2 peptide. Here's NBS1 around. And here would be just interactions. I think that's important, but it's basically, interestingly, the same site where NBS1 binds on one side. We see now TRF2 on the other side. Now, why is this interesting? Because when we have MR alone, so here, sorry, I think this is, this is MR here, so without the NBS1 part, we see basically TRF2 on both sides on the complex, and with MRN, we see it only on one side, whereas the other side is still bound by this NBS1. So this NBS1 prevents binding of TRF2 to one side of the complex, and if you do a superposition, you see why, because they populate basically the same binding pocket, but NBS1 seems to be stronger in this case because, you know, it's bound really in cis with a very high, let's say, avidity, intrinsic avidity here. And this is just a movie, again, where this is kind of summarized. So basically, we kind of load the complex, we clamp down the DNA with, by closing these coils. Here would be the active site. Here's an ADP. We see beryllium fluoride. This has not been yet added to this video, but we see it in the density. Here's this NBS1 site that locks this kind of complex down to some extent. And then we basically see then this TRF2 site on the other side of the complex and locks this side down. So basically, why is this significant? Because how does TRF2 inhibit this machine to prevent activity? And so we haven't really found any influence of ATP hydrolysis. I mean, there's a little bit change, but, you know, I think it's not really significant. What we think happens is that this complex is loaded at the end, forms this kind of sensing state, and then TRF2 blocks one side of NBS1 and of MRN, and NBS1 blocks the other side. And this locks both Rad50 binding sites, and then CtIP, which is the activator, it would also bind to this site, cannot basically attend. And this would basically lead to a one-to-one complex, let's say one MRN complex to one TRF2 monomer here, so there could be another one on this side, but in principle, let's say one. And this would also make sense with other studies, because the Chang lab, they found that TRF2 has like a binding site also for the N-terminal part of NBS1, and NBS1 is only bound in one copy here. So there could be an additional interaction here, basically locking this down in a way at telomeres, which led us to propose maybe there is some function of this complex at telomeres and not at TRF2 is not just getting rid of this complex. So there seems to be a function. You just prevent maybe ATM activation. You prevent further cleavage of DNA through like shutting out CtIP, but there could be a function of this tethering. So with that, I would like to come to the last part here where I show you a little bit what's going on here, because there's another end here that is very interesting. This so-called syncoc region. There's a zinc ion here bound. And as I mentioned, this is a large gate for DNA ends. However, there is something else going on, because in one of our structures, in the fungal one, we found a very robust interaction, a density here. So this gray one, hope you see this, or maybe I use this. Let me see if it's better. Sorry, better pointer. So you see this kind of density. So this would be the one complex, and then you see another density. Now, what is this density? It's actually the same complex, just interacting here again. So it makes kind of dimers through the sync-mediated interactions. Now, why is this interesting? Because if we, let's say, zoom out a little bit, and make like a huge AlphaFold exercise, and then you make two of these complexes through this interaction, you get these gigantic structures here with the two heads separated about by, I think, 1,000 angstroms or something, 120 nanometers, 1,200 angstroms, roughly like this. And we can actually replicate these structures in some conditions on our cryo-EM sheet. So we find, in some cases, these very weird patterns. And what's actually happening is that we get clustering of these heads here. So they form then these stripes. And then these coil regions, they form these linkages here. And of course, in the cell, you would not get such a structure. You would probably get a very tiny piece of it, but they could be very interesting, for instance, in meiosis, where MRN has been shown to play, at least in yeast, and very important roles in maybe pre-structuring synaptonemal complexes or these meiotic break sites. So this clustering has been actually observed before for for 2 decades, basically. So here's like one of our crystal structures where you see a DNA and you see clustering, and then Peter Jager basically had a very nice story about these interfaces, and that actually this clustering actually happens. Also in the eukaryotic complexes, this interaction here between the coils, this is a little bit like maybe these liquid condensate-like things. We have like an exposed phenylalanine that binds into a tiny pocket. We also get a crystal. We have a crystal structure, and you find these very long sheets here that can form. So quite a lot of order. I'm almost done. And these are all pictures from the literature where people find these huge aggregates basically forming at DNA break sites. And so this was a section on tomography, I think the title was, but nobody had shown a tomogram yet. So I show one picture, sorry. So this is a tomogram. I don't have a movie here, so but we basically recorded our tomograms on these structures here, and at the moment we try to basically do some sort of segmentation-based carving out of these heads, and then to hopefully process them. But that's very challenging. I can only tell you this. All right, so I would, I think this is what I talked. So maybe I go to this slide here. Thank all my wonderful people who did a lot of work over time, alumni and the current people with the human structure done by Yilan, Phyllis, and Hengjun, and the bacterial one by Anna. Thank you very much for your attention.
Congratulations, it's a wonderful structure. So the ATP followed by BEV allow you to capture this state. What do you think is happening? You tried the ATP BEV before, right?
Yeah, so I think that's a very good question. So we have also ATP alone and without BEV. So this was our initial work, and there we didn't get a nice distribution, so we never worked on it. But we have a low resolution structure, and if you do a lot of processing and zooming in, what we see is we get an ADP and probably a released phosphate that is still bound. I think that's what's arrested here. So one probably need to get this phosphate release, and then maybe you get this active conformation.
Conformational change, you mean active conformation.
Yeah, so how do you get this active conformation? We have no idea actually, because in bacteria I think it's pretty clear. So this Mre11 will probably is very loosely bound to each other, so you can easily imagine this switch. But the eukaryotic one is so stable, we don't know how you get from this state to the other. We see it's a little bit mobile, yeah?
So that's my second question. You have the NBS1 binding, right? Without NBS1, you see a bit more That's
No.
for MR. No, you still don't get to the cut.
No, yeah, that's a very good point, but the Mre11 itself is very stable interacting. Yeah. So I hope we're not entering in this SMC field, yeah, where people make careers out of gates all over the place, right? So I kind of have a déjà vu almost, yeah. But we'll see which gate opens, yeah.
So I was fascinated by this topological filter functionality. And then I thought, does that help to choose which strand to nick? Because it nicks apparently.
Yeah, it's exactly. Yeah. So this is a good question, and so basically, maybe I go back to one picture here. So the question is, this, although it's a diamond, will nick one strand here, and it's always the same, and it depends where the end is, right? It's the 3' or the 5' strand. So from our current models, the 5' strand needs to be, let's say, the 5' strand nicking needs to be in a way that this part faces inwards from the end. So here would be the break, and here would be the continuous DNA. How this is achieved in the bacterial system, we really don't know, right? Because it could be either symmetric, right? So I think maybe there's something more happening to this coil loading and some pre-interactions that maybe, yeah, we really don't know. In this case, we also don't know, but here we have, of course, NGBS1, so the complex is inherently asymmetric. We have CtIP, maybe this comes from the chromatin side and will decide an end for you, right? Decide a polarity for you, who knows? But it's basically an open question.
Yeah.
There was one here.
The resting sensing head, what's signaling to ATM? I mean, you basically have to take that part off the Rad50 and put it onto.
Exactly. Yeah, we don't know.
Okay.
I guess we all struggle assembling these complexes, right?
Do you have a favorite hypothesis?
Well, so
I was curious about that.
yeah, it's really I don't know. I have a picture here. I think clearly. So I think something is going on with this, and even for the CtIP complex. Let's say if this guy moves over here, and then you would have CtIP sitting somewhere here. What happens to this helix? Because sterically it cannot bind anymore. We don't see how that works, right? And maybe it just wobbles around, right? But often, they like to bind something else to get a stable interaction. This we haven't figured out. ATM, the same thing, yeah? So you can definitely model something, but Mre11 is always in the way. So Mre11 has to go away in a way. Then you get something more reasonable to work out. But this would then argue again, maybe you need CtIP for MRN, maybe you need CtIP to move MRN away, and then maybe you get ATM activation. I don't know, but on the other hand, you need ATM's phosphorylation of CtIP from a stable complex again. So it's kind of a bit weird what's going on there. So I don't know. There's something funny going on, but honestly, we don't understand it.
Yeah. Shall we close then? Thank you very much to